All right, we are recording and we have about 10 minutes left before the lecture starts. So if you are watching this video, obviously you're not online or here, so I'm only talking to you watching the video. You're watching the video, it's business as usual. You need to take a screenshot every time I say one of the screen words, and then at the end of this, you need to send in the screenshots and the screen words. The first word for today is coffee. You can take a screenshot right now, and once you've done that, you can go ahead and skip ahead by about 10 minutes. Because like I said, the lecture's not going to start for about 10 minutes, so you're good to go ahead about 9 to 10 minutes from now if you want. Morning. You have a good weekend?
Yes, sir.
Good. Morning. Did you have a good weekend? Just okay. Mine too. Just kind of boring. I hate that when I was younger. Now I pay money for a boring weekend. Bored all over the world. But it does suck.
I already did the lab drive a couple last months in.
You should be good. I mean, I'll announce that officially, but as long as I get it graded, and you haven't completely dropped the ball, and when I grade it, I'll probably say something like, here's your grade, and say you can make up for lost points, you don't need to come in. All right, it's 7:58. Go ahead and start with attendance. As usual, if you're online and I say you're not here, you don't have to do anything because you're not physically here, so you don't need to correct me. Bangura is not here. Case Bolt is here. Tristan is not here. Covington Griggs, not here. Kately, not here. Dow, not here. Hughes, yep, is here. Lebdy is not here. Little is not here. Bankins is not here. Mathis is not here. Rains is not here. Reed is not here. Elise is not here. Thompson is not here. Ian Dewey is not here. And Elkins is not here. As usual, same old stuff. So if you're online, I'm going to be not watching the video, but if you're live with us right now streaming, I'm going to give out words. You're going to write them down and send them to me before 9 a.m. And the last word I'm going to ask you is to ask me a question about the material. So as I'm going through this, you think of a question you could ask me, jot it down. It doesn't have to be a question that you necessarily have. It could be maybe you just really understood everything and want to give me a good question that I could use on a quiz or an exam. Think about it that way if you want. Anyway, it's 8 o'clock. Let's get started. Need to make some adjustments. We're not going to do announcements and reminders right away. Let's back up to the last thing we talked about. We stopped, the last thing, whoops, what's going on here? Excuse me. Interesting. Is this not working? Bear with me, guys, I apologize. We'll back up this way, maybe that'll work. Yeah, here we go, okay. So this is the last thing we were talking about. We talked about these proteins that pull cholesterol out of your bloodstream. This isn't important. So if you're studying, as a matter of fact, I know this picture's not from your textbook. I don't even know if this example is from your textbook. So you don't need to know anything about the example. I'm not going to ask you about LDL receptors or diseases or any of this. This is just an example to remind you of incomplete dominance. So also as a reminder, prior to Friday, we talked about dominance and recessiveness in a very simple, straightforward way. Like the allele was either dominant and dominated over the recessive one, or it was recessive and was dominated by the dominant one, right? It was pretty straightforward. But as of Friday, I told you there's exceptions to those rules. And this is the first exception that we talked about, was this idea of incomplete dominance, where it's like a merger of the two phenotypes. So the homozygous dominant, and I'll just use the letter, oh, right there, it's already there, H. For any situation when it comes to incomplete dominance, not just for this cholesterol thing, the homozygous dominant has the dominant, obviously, phenotype. The homozygous recessive has the recessive phenotype. But in this case, this is where it makes it different. The heterozygous genotype has a merger of the two phenotypes. So on the exam, for this portion of the exam anyway, a lot of the questions are going to be like me saying, all right, I mated this thing with this thing. Here were the results. Is this an example of complete dominance, incomplete dominance, or some of the other ones that I'm about to teach you? So that's a lot of what the questions are, how the questions are going to go. So remember, if it's a merger of the two phenotypes, a blending of the two phenotypes, that is incomplete dominance. So for example, again, and I used this example on Friday, if I say a blue alien mates with a yellow alien, a purebred blue alien, right, so homozygous dominant, purebred mates with a purebred yellow alien, so homozygous recessive, and all their offspring are heterozygous in green, then you would say, oh yeah, that's incomplete dominance, because when you mix yellow and blue, you get green, right? So think about it that way. That being said, let's move on to the next one, and I'll go ahead and tell you, or first of all, I'll remind you, and then I'll tell you something. I'm reminding you that this PowerPoint is based off of the old textbook. So again, some of the examples are different. Some of the order in which I'm covering things are a little bit different. So that being said, I'll go ahead and tell you now, even though this says ABO blood groups, I'll go ahead and tell you I'm not going to probably ask you any questions about ABO blood groups. ABO blood groups are an example of what we call codominance. So keep that in the back of your mind. What I'm about to teach you is a different type of dominance. It's not incomplete dominance, it is codominance. So let's jump into it. ABO blood groups involve 3 different alleles of a single gene. And I think this is why this is an interesting example, because so far also, we've only talked about 2 alleles, right? Everything we've talked about either had a dominant allele or a recessive allele. And in this situation, they have 3 alleles. But that being said, even though there are 3 alleles, I'm not going to get that complicated in the exam. There's not going to be anything where there's 3 alleles. When we're talking about complete dominance or incomplete dominance or codominance, I'm going to keep it simple, and we're only going to be dealing with things that have 2 alleles. But yes, we will be discussing this. Your first word for attendance is to tell me. This is I guess it's not a word, it's a number. The ABO blood groups involve how many alleles? Again, I'll remind you, if you're in person, you don't have to send these words, but you can if you want extra credit. If you're online, you have to send the words to get any credit, any of the extra credit, and to be considered present. And if you're watching the video, you have to send the words, and you have to send the screenshot of when the word was said. Yeah, and also if you're online, don't forget you need to send them by 9 a.m. And also, again, as a reminder, you need to be online. Don't let your buddy send you the words and then you, because they were paying attention and you send them in. Someone tried that once. Don't do that, please. Anyway, back to this. So there's various combinations. I'm sure you guys have heard of like, oh, I'm O positive, O negative, AB positive, whatever. I'm sure you've heard that. And this is half of what we're going to talk about today here in this section. So we're not going to talk about the positive or the negative, the plus or the minus. You can look that up for independent work if you want. We're going to deal strictly with the letters. A, B, AB, or O. Those are the four blood groups that we're going to talk about. They actually refer to carbohydrates. Like I said, I'm probably not going to ask you any questions about this on the exam. If I do, I'm definitely not going to ask you that. But just so you know, this is where they got their name. They refer to these carbohydrates that are on the surface of the blood cells. This is a picture that's not from your textbook. So even though there's no reason to study this, because I'm not going to ask you any questions about this, but if for some reason you wanted to study it, you can, and you would have to find it in the PowerPoint, not in the textbook. So again, these letters stand for these carbohydrates, right? So if somebody has the A allele, you can see right there that I with a big A next to it, right? That's kind of blurry, it's kind of hard to see. But anyway, if they're homozygous dominant with an A, or even heterozygous, you can see here, no matter what, as long as they have that A there, they have carbohydrate A on the outside of their blood cell. And I'm not even going to get into that. You can look into that for independent work if you want. What are these carbohydrates? I'm sure they're not actually named A and B. What are they actually named? What's their purpose? Things like that. But anyway, if you have carbohydrate A on the outside of your blood, that means your blood is anti-B, which means if you need blood, you cannot get blood from anybody that came from a B, because your blood is the anti-B. You can't even get blood from somebody who's AB, because even though you can see down here, they have both carbohydrates, it doesn't matter. Your blood is hateful, so to speak, right? It doesn't matter that AB has A on it. It just hates B. And of course, the opposite can be said for B. It has carbohydrate B on the outside, therefore it's anti-A. So it can't take A, it can't take AB. They can only take A or O, and I'll talk about that here in a second. AB, this is somebody who has both carbohydrates. They are what's called a universal receiver, because they've got both carbohydrates, so they don't care. Any kind of blood is good for them. And meanwhile, I'm kind of the opposite of that, which is what I am. O. O doesn't have any carbohydrates. So it sucks for me, because if you're O, that means you're anti-everything. You're anti-A, you're anti-B, and say you've got the picky blood. You can only take other O blood. On the flip side, though, O blood is the universal donor, because anybody can take O. Anyway, before I move forward, I'll remind you, none of this is going to be on the exam. Well, most likely none of it's going to be on the exam. If it is, it'll be something very basic. I only included it because a lot of times people hear about these blood groups and have no idea what they mean. So now you know. Anyway, you can read all about it if you want. But more importantly, again, you can read all this if you want. It is an interesting example because, again, it uses 3 alleles, and this is the first time we've dealt with 3 different alleles. But again, for the exam, I'm not going to use anything that has 3 alleles. So let's get into what's more important as far as the exam is concerned, is this slide right here. These are just some non-textbook examples, and I made most of them up. Actually, this last one is a real example, but it doesn't matter. Actually, so is the cow. But anyway, imagine, and this is how the test might go. Imagine you mix a black dog with a white dog, and then the offspring are black dogs with white spots. That would be codominance. As a reminder, to go back to what we were talking about earlier, if you mix a black dog and a white dog and you got a gray dog, which is like a mixture of the two, that would be incomplete dominance. Another example, and again, these are just examples, so if you're studying, don't feel like you need to memorize these examples. Another example is a red cow and a white cow. Would give you a white cow with red spots, or a red cow with white spots, however you want to say it. The point is, it's not a mixture of the 2 phenotypes. It's both phenotypes showing up independently. And again, one last example for this is a red flower and a white flower. Remember, that's also the example we used in incomplete dominance. We said, all right, we mix this red flower and a white flower, and we get a pink flower, because that's a blend of the 2, right? That would be incomplete dominance. Now, in this situation, when you have a red flower and white flower, we're talking about codominance, where both phenotypes show up individually. This would be a red flower with white spots, or a white flower with red spots, however you want to say it. So again, for the exam, you probably have at least 2 questions for each of these. One of which will be me giving you something like this, like, we made it this thing with this thing, here were the results. What kind of dominance is this? And then there'll be another question when I tell you what kind of dominance it is, and I say, all right, this is an example of incomplete dominance or codominance or whatever the case may be. And we mix this thing and this thing. What are the chances of their offspring being that thing? And I created a document on Friday. I think it was Friday, either that or sometime over the weekend. Anyway, I created an extra credit document. I shared it on Facebook. I don't think I shared it on the announcements. Anyway, it gives you a lot of practice where you can do these things. Where I'm like, all right, here we mix this thing and this thing. What are the probabilities of getting that thing? So if you want and you didn't see that, email me and let me know. I can send you that document. It's worth what I think at least 10 points of extra credit. And again, it'll help you prepare for the exam. And also, since you have to use a Punnett square to do it, those extra credit points, actually, I think I did post it for this reason. Anyway, these extra credit points can be applied to the last week's lab. And some of you really need it. Some of you didn't do the damn, pardon my French, but you didn't do the pre-lab in time, so you only got half credit. Some of you didn't even show up at lab, so you're only getting half credit. So, take advantage of those points. Anyway, the next word for attendance is I'm going to give you 3 colors of cows, 2 of which we talked about, 1 of which we did not. Tell me the one that we did not talk about. Brown cow, red cow, white cow. Of those 3, which color cow did we not discuss on this slide? All right, any questions about codominance or incomplete dominance for that matter? All right, in my opinion, these 2 are the hardest to distinguish from each other. I mean, they're very different and they're very simple concepts, but the names are so similar, right? Codominance versus incomplete dominance. And even the way you try to explain it verbally, even that is kind of confusing, right? So like when I say incomplete dominance, it's a blend of the 2 phenotypes, and codominance is both phenotypes showing up at once. And you could almost argue those definitions could be interchanged almost. But anyway, just make sure you understand it and let me know if you don't. Any questions? Great. All right, before we get into the next variation on Mendel's law, let me do announcements and reminders. So the DNA lab is this week. It's 100% virtual, right? So this is why one of the reasons why I didn't care about the pre-lab, because everybody, you're doing it on your own. So if you're not prepared, you're not holding the class back. So that's one of the reasons I don't care. Anyway, because it is all virtual, and I received this question earlier, you might not have to come to lab. So for example, I know one student, because they asked, they said they've already done the lab. And as soon as I can, once I'm caught up with some more time-sensitive stuff, I will grade their lab. And if it looks good, I will tell them, hey, here's your grade, and you do not have to come to lab this week. So there's a possibility you might not have to come to lab this week. I will let you specifically know. So if you don't hear anything from me, come to lab this week. But again, if you get it done before Wednesday, there is a chance, if I can get it graded in time, that I will tell you specifically you do not have to come to lab. But unless you hear from me, you need to come to lab. Any questions about lab this week? All right. So from now on, because attendance is so horrible and grades are so horrible and due dates are so horrible, from now on I'm offering double the extra credit to attend in person. So already it's been, if you're online, you get 1 point of extra credit. If you're in person, you get 2 points of extra credit. But it's been so bad, starting on Wednesday, if you show up, you will get 4 points of extra credit. And if you're online, you still get 1. So I'm just trying to bribe you to come in, because a lot of you are just like missing things that I like. Like, how did you miss the exam? I said it every time we met. Like, hey, don't forget. I had an announcement reminder, right? This is the due date for the exam. This is the due date for the exam. This is the due date for the exam. Anyway, yeah, so there's the bribe. Double the extra credit to attend in person. Again, exam 2 is open. I keep saying this, but it's the same thing. If you haven't, actually it doesn't matter. Whatever your grade was, whether it be a 0, nobody got 100. So if you got a 0 or a 98, whatever it may be, you can retake the exam as many times as you want, open book, not timed. Once you're satisfied with your grade, email me, say, hey, can you change my grade? And then I'll go in and I'll take your highest grade and your first grade, and I'll average the two. Don't forget you have to email me, otherwise I won't know to change it. It won't happen automatically. Any questions about anything so far? Okay, again, the last day to drop this course is sometime this month. By Wednesday, maybe if I can remember, I'll look it up and give you the exact date. Some of you should either drop it or start turning things around. So now we have about 7 weeks left to get 100 points of independent work. Some of you haven't done a single paper. I've been begging you, please send me one paper, at least one, so you know how I grade. Because then if you know how I grade, then you'll know what you're getting into. You're like, okay, now that I know, now I'm going to procrastinate, wait till the very end and turn in the rest. But some of you are not going to do that, and you're going to turn in a bunch of work on the last weekend and think, all right, I did it. And then it might turn out that you did not do it at all and got 0 points because you missed some kind of big thing that you would have known about had you turned in a paper. So turn in a paper. Remember, your paper can be 1 page or 100 pages, or one sentence or 100 pages, whatever. It doesn't matter. There is no maximum or minimum. Any questions about anything so far? All right, as a reminder, I'm here to help. So before you turn anything in, whether it be independent work or pre-lab or anything, if you want, just meet me online and be, hey, can you look this over very quickly to make sure I didn't miss anything big? I'm here to help. Or again, for any assignment that we've done this semester, if you meet me online to get full or get detailed feedback, I will also give you a 5% boost to that grade, including exam 1. And then finally, like I've been saying, office hours for the Friday before Easter and the Monday after Easter and lecture will be 100% online. So we will not be in person on Friday the 18th or Monday the 21st of April. Any questions about announcements, reminders? You guys are nice and easy. All right. So again, we are talking about variations on Mendel's laws because not everything is as straightforward as I first taught you, where we've got one dominant allele or one recessive allele, and the dominant always beats out the recessive. It's not always that simple, right? We finished talking about incomplete dominance. We finished talking about codominance. Now we're going to talk about something else called pleiotropy. This one's a little bit harder, a little bit. The concept, I think, is just as easy, but the name isn't as easy, right? Incomplete dominance really kind of describes what happens in that situation. Codominance really describes what happens in that situation. Pleiotropy, that's not as easy. But let me give you an example. And I'm not going to ask you about this on this example. Well, in this context, when it comes to genetics or pleiotropy, I will not be talking about sickle cell disease. But you'll see we're actually going to use this disease as an example when we talk about mutations later and when we talk about natural selection later. So sickle cell disease will be an example we use a lot. But anyway, sickle cell disease is a great example of pleiotropy. And I'll tell you why, but first let's talk about it a little bit. What happens is, if you have sickle cell disease, this results in an abnormal hemoglobin protein. Not that you need to know what that means, but basically what that means is you've got these red blood cells, there's big proteins in them, big and important. And when you have this disease, as you're going to learn later, there's a mutation in the gene that codes for the protein. So therefore, the protein has a wrong shape. Because it's such a big protein, not only does the protein have the wrong shape, you can see in that picture over to the right, the blood cell also has a weird shape. And that might not sound like a big deal, but it is, right? You need your blood cells to be the correct shape. So this is what it leads to. So again, if you're homozygous for sickle cell, if you're homozygous for the sickle cell allele, that means you have the abnormal hemoglobin protein. Therefore, you have this abnormal shape of the blood cell, and therefore you have all these symptoms. And that is the key. It's all these symptoms. Now remember, this is just an example, so I'm not going to ask you about the symptoms of sickle cell disease. But here we go, what you need to know for the exam. In pleiotropy, we're still dealing with one gene, right? We're still dealing with 2 different alleles of that one gene. However, in this case, it's not one gene, one character, right? And this one gene leads to all kinds of different things, right? In this case, it leads to weakness, pain, organ damage, paralysis. So anytime you have one gene that affects multiple characters, that is an example of pleiotropy. So unlike everything we've just learned, where like there's the one gene in charge of your chin dimple, or the one gene in charge of your eye color, or the one gene in charge of the color of the cow, right? In this case, you have one gene in many different characters. That is pleiotropy. That's what you need to know for the exam. So again, I might say, we made it this thing, and here, actually, you know what? I take that back. For the exam, what I'll do is instead of saying, we made it this thing, and here was the results, I'll say something like, I give an example, not this example, but I'll give an example of pleiotropy. It might be made up, it might be real, but it'll be something like, you're a doctor and you have this patient. You looked at their genome and maybe you found out that they have this one mutated gene. But because of this one mutated gene, they have this, and this. And that's the key to understanding pleiotropy. If I say one gene and then I list a bunch of stuff, that's pleiotropy. So any questions about this concept of pleiotropy? Okay, nice and easy. We'll move forward. So now let's talk about the opposite of pleiotropy. And unfortunately, it also starts with P, which makes it a little bit confusing. But the opposite, if you want to call it that, of pleiotropy is polygenic inheritance. So instead of having one gene affecting one phenotype or one character, now you have a lot, more than two genes, at least two genes or more, that affect a single phenotype. So instead of again, using the chin dimple as an example, instead of having one gene for that one character, even though this isn't the way, it would be as if you had multiple genes all determining if you had a chin dimple or not, or how deep your chin dimple is, right? But that's not how it works, it's just an example. So yes, polygenic inheritance is, again, just one phenotype with many genes. Here's the key for the exam anyway, because this is probably how the question's gonna go. If you were to plot the phenotypes on a bar graph, you would get a bell curve. Let me give you an example of what I mean before I show you the next slide, which is a great example. If you were to plot a bar graph, and some of you did do this for the last lab, of chin dimples, right? Recessive or not recessive, right? You might, I won't even ask, I'll just do it. Let's say that was recessive, and then you had a bar here for dominant, right? If you were to graph the different phenotypes for chin dimple, you would only have two bars, right? Same thing for eye color. If you were to graph the eye color thing, you would only have two bars. Well, yeah, in that situation, you only have two bars, right? Blue or not blue. Chin dimple, you either have it or you don't, right? Two bars. But if you were to graph the phenotype of something that is influenced by polygenic inheritance, it would be a bell curve. And again, the reason I'm harping on this so much is because I wrote the exam, and if I remember correctly, that is the wording like that. The wording for the question does involve bell curve. So if you see anything with a bell curve, that's the answer. So can you guys think about it? Think about, all right, let's see, before I move forward, I'm gonna see if anyone can guess this. Is there any phenotype you can think of that doesn't come in just two varieties, right? So again, two varieties, you either have a widow's peak or you don't. Chin dimples or you don't. Freckles or you don't. Blue eyes or not blue eyes. Hitchhiker's thumb or straight thumb, right? Those are only two varieties. Is there a phenotype that you can think of? It's a really big one and a really common one too, that comes in more than two varieties. I'll give you a hint if you guys don't mind. If you're online, sorry. Why don't you guys stand up and look at each other. We'll all look at each other. It's kind of a bad example because you guys are kind of similar, but all right. Probably a good stretch too. Anyway, any guesses yet? Height? Height, yes, that's the great example. Imagine if every human on Earth was either, I don't know, short, medium, or tall. That would be, that might be your graph, right? But it's not. This is oversimplified, right? So it's not like everybody's either, I don't know, I'm just gonna make some numbers up. It's not like everybody's either 5'5" or I don't know, 5'8", 6'5", and so on and so forth, right? And we know that even this is oversimplified. In all actuality, there's everything, every little thing in between. We're talking about centimeters in between people. So this is an oversimplified example. Even this is a Punnett square. That thing, that big, huge, confusing thing. that's a Punnett square dealing with only 3 alleles too, right? So, we're only dealing with 3 alleles in this example, but there's actually a lot more involved in height. It's not just 3 alleles. So imagine how confusing, or not confusing necessarily, but imagine how complicated it must be if you're actually dealing with more than 3 alleles. Because again, this is oversimplified and it's only dealing with 3 alleles in the example. Another thing to note, and I'll talk about this on the next slide, or the next concept, but also in all actuality, again, this is just an example, height is not just about genes. It also is influenced by other things like, your diet as a child and things like that. But height is still a pretty good example of polygenic inheritance because, again, it's more than one gene influencing it, right? And that's what you need to know for polygenic inheritance. And for any of these, if you want, for independent work, let me back it up one step, or just move forward one step. I gave you some examples of incomplete dominance. For independent work, look up some other real examples of incomplete dominance that I didn't say. I gave you one example, well, I gave you a few examples of codominance. For independent work, look for some real examples of codominance. I gave you one example of that should be polygenic, not genetic. Anyway, I gave you one example of polygenic inheritance, which was sickle cell. What are some other examples of polygenic inheritance? And here, pleiotropy with sickle cell. I gave you one example of pleiotropy with sickle cell. Feel free to look up more for independent work. And the polygenic inheritance, I gave you one example, which was height. So you can look up others for independent work. Anyway, let's talk about epigenetics. I already kind of hinted at this when we were talking about height. So basically what this boils down to is when we're talking about epigenetics, it's like, all right, not everything's strictly genetic. Sometimes it's also things are influenced by your environment. I don't know if you've heard the term nature versus nurture. Well, epigenetics would be like, it's not nature versus nurture. In this case, it's both. They both influence things. So there's a lot of debate about this. And if you're into it, you could write about that for independent work. Again, thinking about things like, I don't know, homosexual, I'm not necessarily saying, well, sure. Sexual preferences, right? Like some people say it's genetic, some people say it's not. You can look that up for independent work. There's a lot of things where some people argue it's nature versus nurture. Some people argue it's epigenetics. It's a mixture of both. Some people argue it's nature or nurture. So these are things you could look at. The good example that the old textbook gave, and I think yours does too, are identical twins. Do any of you guys know, like, spend time with identical twins? You do? Can you tell them apart? Yeah, right, because they are different, right? Now, if I met them, I probably couldn't tell them apart. But after a while, you get to know the difference. And they are, right? They're not just different because they're different people, but because even if they live in the same house and they mostly have the same environment, it's not exactly the same environment, right? There are some differences, and eventually those differences change the way they appear and everything else, not just the visual phenotypes, but also the invisible phenotypes. In the next slides, I'm going to explain how that happens. But for now, I'll just leave it at that. That's a good example. So in general, that's the key part, not always, but in general, it is only the genetic influences that are inherited, but the effects of the environment are usually not passed on to the next generation. So for example, if you smoke a lot, and you get cancer because you mutated some genes, mutated some DNA for the cells that are in your lungs, right? You mutated them, therefore you got cancer. Let's say they cured it, it never spread, and that's the end of it, right? Your offspring should be okay based on what we know mostly about genetics, because you didn't mutate your sperm or egg, right? You didn't mutate your gametes, you only mutated your lung cells. Therefore, you're not going to pass that along. That's how we've known it for a long time, but now we're starting to understand that's not necessarily true. In recent years, biologists have begun to recognize the importance of epigenetic inheritance, which is the transmission of traits by mechanisms not directly involving the DNA sequence. I'll give you an example like this. Think of your chromosomes as a book, right? So let's look at one book, right? You have this one book out of 23 pairs, right? Looking at this one book, and remember, a chromosome is a set of instructions on how to build proteins. So what we're saying here is we're not changing the book. We're not changing the writing in the book, right? That would be changing the DNA sequence. We're not doing that. What I'm saying is, imagine for some reason your book, the page got stuck together. I don't know why, I'm not gonna ask. But for whatever reason, one of your pages got stuck together. Your brother, your identical twin brother, their page is not stuck together. So when it comes time for your body to build this specific protein, when they go to your book, can't read the code, right? We didn't change the code, but you stuck the pages together, so now we can't read the code, and that protein does not get built. Meanwhile, your identical twin brother, who has the same exact book with all the same exact words, we can read the directions because they didn't stick the pages together. Hope that makes sense. The point here is we didn't change the words in the book. They're still exactly the same. What we've changed is how easily, or if at all, you can read the words in the book. Another example would be, if you ever put like a book down, you're reading it and you don't have a bookmark, so you just put it upside down, and what happens? The book kind of like wants to stay that way, right? So that's kind of the same situation too. If you had a book like that for a week, and then you fixed it and picked it up, most likely when you open up that book, it's going to open up that page, right? Because it's been like that for a week. So you've done something to your book where you didn't change the code, the words inside, but you've made it to where now it's really, really easy to read that page. It's kind of like the opposite of what I was saying before, where the pages stuck together. So in that case, whatever protein directions to make the proteins that are on that page, that's getting read a lot. You're making a lot of those proteins. Not because you changed the words, but because you changed how easy or hard it was to read the words in that book. So that is epigenetics. So again, using that analogy, epigenetics is like you do something in your life that causes your book to be set upside down or the pages stuck together. And because of that, because of those changes, sometimes you can also pass that along in your gametes. And therefore, some of the things you do bad or good to yourself can sometimes be passed along to your children. Anyway, and I didn't give you any examples of that. So for independent work, look up some real life examples of epigenetic inheritance. Actually, I'm about to give you an example. This is basically everything I just said. I'm not gonna read this. This is what I said. This is the example I gave, except it talks about it in more technical terms. We didn't change the A's and the T's and the C's and the G's, but let me talk about this first bullet point. If you remember when I first introduced DNA, I said that it's, DNA is wrapped around these proteins, and those proteins are called histones, and there's 8 of them, and for every 8 that are wrapped around with a DNA, we call that a nucleosome. And I said later in the semester, this is going to be important, and this is where it's important. So now instead of me just giving the analogy and saying, yeah, your pages are stuck together, or you put your book upside down, now I'm telling you how it actually happens. Your DNA is wound around those proteins, and there's things you can do in your life that makes it looser around those proteins, or maybe even tighter. And that matters because, as you're going to learn in the next chapter, there's cellular machinery that clamps onto your DNA and reads it. So if you make it harder for the DNA machinery to read it, then you're not going to build those proteins, or not build them as often. Or the opposite, if you loosen it up, that machinery can clamp onto the DNA and it can read it easier, and therefore you're making that protein a little bit more. So that's it. That's epigenetic inheritance. And I guess that's not really an example. That's more of an explanation of how it works. I need to fix this. So yeah, for independent work, again, you might want to look up some real life examples of epigenetics. And you'll probably find a lot of stuff. There's some stuff where people are hypothesizing that this is controlled by epigenetics or that, and they haven't figured it out yet. They're still working on it. Anyway, the next word for attendance is, at the bottom of the screen, there is a list of DNA bases. And what is the last one? So it starts with an A, and then it goes to a T, then it goes to blah, blah, blah. What is the last letter listed? Is the next word for attendance. And again, if you're watching the video, so not the live feed, but if you're watching the video at a later time, don't forget to take your screenshots. All right, a little bit more about epigenetics. And I've already said this, and now it's just in writing. Epigenetic modifications and the changes that result from it, right? So again, like whether you stuck your page together or opened your book up, whatever, can be carried on to the next generation, like I said. But the good news is, as far as we know, Unlike alterations to the DNA sequence, so instead of, again, unlike changing the actual words in the book, these can be undone, right? These changes can be undone, which makes it very interesting. One thing that I don't think your book talks about, and I know this old textbook didn't talk about, that you can look into if you want for independent work, is something called omnigenetics. We don't talk about that, but this is a new up and coming thing. Omnigenetics. And this is a school of thought where they say every single protein that your body makes in some way or another is going to influence every other protein. So that's what omni means. It means like every time you express one protein, it's going to affect every other expression of a protein, whether it be major or minor. They're all interconnected. And again, you can look that up for independent work if you want. Any questions about epigenetics? We'll talk about that. Okay, let's make things more complicated. So again, we know about Mendel. The first thing I taught you about was the simple complete dominance, right? Dominant or recessive alleles. Dominant or recessive phenotype, nothing in between, right? That's what we started with. Then we just now got finished talking about those variations on Mendel's laws. Now we're going to talk about something even different. Well, yeah, we'll get to that. Sorry, I jumped ahead. What I was about to get into was talk about these sex-linked genes, which have a different inheritance pattern. We'll obviously cover that when we get there. So hopefully I can make yet a third document between now and the exam and share that document with you so you can have even more practice where I say this thing mated with this thing, what are the chances of their offspring being this? Right? So far, none of those examples, none of those that I did I haven't done any of them with sex genes. So we'll talk about that when we get there. But first, let's talk about the chromosomal basis of inheritance. Before I jump into it, I'll go ahead and tell you, again, I'm teaching out of the textbook. So what I'm about to tell you, I've already told you. So even as we were talking about these alleles previously, I was already talking about the chromosomes. So we weren't just looking at the alleles, like you've got this allele versus that allele. I've already mentioned the fact that these alleles are on specific chromosomes, but this is what we're going to talk about. I'm going to go through it very quickly because, again, I've already mentioned this. I've mentioned it throughout the discussion, and now I'm going to officially bring it up. The chromosome theory of inheritance states that genes are located at specific positions on chromosomes. So you already know that, right? So again, using some example of, I don't know, having crooked teeth, and this is not how it works, but if having crooked teeth is controlled by one gene found in the middle of chromosome 22, for me, then it would be like that for every other human, right? That gene is located on that chromosome at that spot for every human that doesn't have anything wrong with their chromosomes. That is what chromosome theory of inheritance states. That's part of it, right? So there's some very specific genes at specific locations. And what I'm really about to get at, and what I've kind of talked about throughout this discussion, it's the behavior of the chromosomes during meiosis and fertilization that accounts for the inheritance patterns. So basically, I'm going to go through this very quickly. It's the same thing we just said that I've been saying, except now the old textbook, and I don't remember if your new textbook does it or not, but your old textbook brings back an old example, but now instead of just having the alleles, they've also drawn little chromosomes to show that the chromosomes are on alleles. So that's it. We're not going to get into it that much. The point is, again, these alleles are on specific chromosomes. And that was a very important discussion back when we talked about independent assortment. Remember when we were saying when the chromosomes line up in the middle for meiosis, metaphase of meiosis, it's not like all the mom chromosomes are on one side and all the dad chromosomes are on the other, right? It's all random. That's all this is getting at. So this looks really complicated. If it's overwhelming, don't worry about it, because I'm probably not going to ask you any questions about the chromosomal theory of inheritance. The point is that I just want you to remember in the back of your mind, not even for the exam, all these things, when we keep talking about these alleles, which allele do you get? Do you get your mom's allele, or do you get your dad's allele? You gotta look at the bigger picture, because that allele that you're talking about, it was on a whole chromosome. So did you get your mom's whole chromosome, or did you get your dad's whole chromosome? And when you create sperm or egg, are you giving, and they separate, right? But that's a better way of saying it. When you go through meiosis and you produce sperm or egg, and they segregate, right? Was this particular sperm getting your dad's chromosome or your mom's chromosome? Or if we're zooming in and talking about a specific gene that's on that chromosome, are you getting your mom's gene or your dad's gene? That's all this is saying. It's a good reminder. Anyway, any questions about that? All right, let's talk about linked genes. There might be like one question about this, and I'll go ahead and give you the answer before we even talk about it. So when we talk about, remember we talked about independent assortment, which was saying, again, when you have two different genes, they're not necessarily going to travel together, right? So if you got your mom's gene for chin dimple, that doesn't mean you got your mom's gene for eyes, right? Because they're on different chromosomes. You could have, it's possible, or it could be that you got your mom's chin dimple gene and your dad's eye gene, or maybe your dad's chin dimple gene and your mom's eye gene, right? We don't know. Those, all right, so those are, again, genes that are not on different chromosomes, or the genes that are on different chromosomes, they're not linked to each other. So you can get any combination.
So it's really just random.
It is, yeah, exactly. So again, if you were to think about three genes, right, freckles, chin dimple, and eye, right, it is possible that you would get all three from mom, I'm sorry, I was talking about your sperm in your case. It is possible if you were to choose one specific sperm and look at it, that they got all three of those genes from your mom. And then maybe another one has all three from your dad. Maybe another one has mom, mom, dad. Maybe the other one has dad, dad, mom, right? It's all random because they're on different chromosomes. Now this is the exception to the rule. What I'm about to teach you is called linked genes. So before I even get into it, again, oh no, it's on here, I'll just show you. Linked genes, however, are on the same chromosome, which makes sense, right? Chromosomes are big. So some of these genes, like I was just using that example as chin, dimple, and freckles, those are on different chromosomes, but some of them are on the same chromosome. So say, for example, and I'm making this up, this is not the case. Let me draw this other chromosome here. So here's a pair of homologous chromosomes. Let's say this is the one you got from your mom, this is the one you got from your dad. Let's say this is for eye color. No, we won't need to do gene 1. Let's do gene 2. Let's say gene 2 is for eye color, and I'm completely making this up, and gene 3 is for freckles, right? Come on. Whatever, it's not letting me draw it. If that were the case, in this case, if you did get your mom's eye gene, you probably would get her freckle gene too. Does that make sense? Because if you got her chromosome, then for the most part, you got all of the genes on that chromosome. And the reason I say that for the most part is don't forget there is crossing over, right? So there is a chance that, you know, this section of the chromosome from your mom will swap with this section of the chromosome from your dad, right? So then you'll have the mom chromosome with the bottom chunk of the dad, and the dad chromosome with the bottom chunk of the mom, right? So sometimes they can't get swapped. But if there was no such thing as crossing over, which for this discussion we're going to pretend there's not, there's no such thing, then any gene that you get from, in this example, any gene that comes on your mom's chromosome, you know, they're all going to go together because they're on the same chromosome. Long story short, as far as the exam is concerned, this means if you see anything, we're talking about a dihybrid cross, and again, to remind you, that just means instead of just talking about just chin dimples or just freckles, dihybrid cross, we're talking about chin dimples and freckles, right? It's when you talk about two things instead of just one. Again, you would expect a 9-3 to 3-1 ratio if you have two, purebreds, and we're talking about dihybrid cross. In this situation, because they're linked, if you see any ratio that's other than 9-3 to 3-1, then you know it's a linked gene. So for example, I don't know, 10 to 3 to 2 to 1, whatever the case may be, right? Point is, and again, that's the Cliff Notes. That doesn't help you actually understand how it works, but for the exam, so I get it. That's all you care about. If you see a ratio that has 4 digits and it's something other than 9-3 to 3-1, that means it's linked genes. So that's what you need to know for the exam. But again, for me, what I want you to know, more importantly, is that the idea that, again, these genes, you got genes on chromosomes. So if you're looking at the mom chromosome, then other than crossing over, you're getting all the genes that came on that mom chromosome. All right, that was pretty complicated. Let's jump into something very easy, sort of. This is a 100 level course. It's a very hot topic right now, so you can definitely get into it for independent work if you want. I'm going to give you the Cliff Notes on sex determination in humans. Many animals, including mammals, have a pair of sex chromosomes designated X and Y that determine the X, or the sex. So, from this point of view, a male is somebody who has an X and a Y chromosome, and a female is somebody who has an X and an X chromosome. You've already seen the table where there's problems with non-disjunction. So the one example you had in lab was X, Y, Y, Y. Then there could also be X, Y, Y, or there could be XO or XXY. Right? There's also, that's a good example. There are some males who have an extra X chromosome, and an X chromosome is a female chromosome. So they have, like, it's a male that has more feminine things about them. So, but yes, for the exam, a male is XY and a female is XX. For independent work, you can look up all the different variations of that. Again, having extra Y chromosomes, extra X chromosomes, missing sex chromosomes. You can write papers on that and tell me about it. Another thing we can do for independent work is tell me how else it can be determined. Because, sometimes, for example, children are born with both genitalia, right? And their genome would also be ambiguous. So then how do you determine their sex? What do you put on their birth certificate? Things like that. Anyway, that's it. Again, as far as the exam is concerned, what you need to know is a male is XY and a female is XX. For independent work, just a whole slew of papers you can write about that, especially right now, because again, it's a hot topic. People like to debate about it, so a lot of people are writing about it. Which reminded me, you have to be careful with your sources. There's a lot of misinformation out there too, especially when it's a hot topic. People like to spread a lot of misinformation about hot topics. Anyway, here's a picture of it. There's an X chromosome, there's a Y chromosome. So we're looking at a male here, right? X chromosome, Y chromosome. Any questions about that? All right, let's start this discussion about sex-linked genes. So again, you already know about dominant, recessive, and pleiotropy, and incomplete dominance, complete dominance, and this is sort of like I don't say it's a variation of Mendel's law, but it does make things a little bit more complicated, and we'll see why. A gene located on a chromosome obviously is a sex-linked gene. So this kind of goes back to what we were saying when we were talking about the chromosomal theory of inheritance, right? So these genes that we talk about, it's not like they're just floating around in your cells. They are attached to a specific chromosome. And these happen to be attached to either an X chromosome or a Y chromosome. And you've got to think about how that must matter, right? Because for a female will never, as far as a female will never have a Y chromosome, right? So therefore, anything found on a Y chromosome, a gene found on a Y chromosome, will never affect a female. Males, on the other hand, will have an X and a Y, and that'll make things a little bit more complicated too, as you're about to see. And I'll give you some examples. Some conditions, including red-green color blindness, hemophilia, and a type of muscular dystrophy, result from sex-linked recessive alleles, and that'll be important here in a second. Before I talk about it, I put a big old X through here, because I'm not going to ask you specific questions about this. But this is a great example of a sex-linked recessive allele, red-green color blindness. It's a common sex-linked disorder caused by a malfunction of light-sensitive cells in the eyes. It's mostly the males that are affected. Heterozygous females have defects too, and the next slide shows a test for it. So I'm not going to wait a long time, but I'm going to ask this question. If no one gets it, I'm going to move forward. But you could answer it based on the information on this slide. Knowing what you see here, what chromosome do you think this disorder is on? The X chromosome or the Y chromosome? That's okay. We'll get there. Anyway, can anybody not read that number? Number
7.
Is it number 7? Yeah.
Yep, it's a 7. If you can't see it, you might have red-green color blindness.
Look what a green is.
Yeah. You might have. It might be a. It might be a bad projector, or it might be red-green color blindness. Anyway, because they're located on sex chromosomes, sex-linked genes exhibit an unusual inheritance pattern, which is again why I'm hoping to make a new document that you can do for extra credit to give you some practice with this. But to answer your question, it is on the X chromosome. The reason we know this is because of this right here. Mostly the males are affected, but females are also affected.
I was going to say that.
Yeah. I didn't want to wait too long, but yeah, the fact that some females are affected tells us it can't be on the Y chromosome. Anyway, you know what? Let's see. What we have here in this first example, is two people being made, we're seeing the mating of two different people. A normal female and what kind of male? That's your last word for attendance. Other than the question, don't forget you have to ask me a question. So the second to last word for attendance is to answer me this question. We're looking on the screen of a mating between a normal female and what kind of male? That's the last word for attendance, other than you asking me a question, because it's 8:49, and that'll be it. So we're done for now. I will be in my office and online for office hours. You guys have a good week.
I came in late.
Oh, yeah, send me an email, please, to remind me. Actually, I'll just do it now. Graves.
Yes, sir.
Oh, yeah, and then you do it, right?
Yeah.